I'll just ask, have people had success accessing the videos either through the ICON website or through the Google video? If you've had success with either of those, could you raise your hands? If you've had problems with both, could you raise your hands? So you haven't been able to access it either way? Have you tried the Google video? And it doesn't work on Google Video either? Really? We can talk after class then. Is there anybody else who's had trouble with both ICON and Google? Okay. We can talk after class if you like them. Great. So you should see, there are a number of sort of practical things we wanted to take care of first before we start talking about the next chapter. You've given me some feedback on the movies that for the most part, one or the other will work. Generally speaking, Macs can't use the ICON video, but they usually can use the Google video. So hopefully one way or the other is available to everybody. We will be making available practice exams and review questions for your preparation for the exam. And just a gentle reminder that if you need accommodations for the exams, please let me know soon and we'll make sure that those accommodations are worked out. I generally give fundamentally unlimited time for the test, so if the accommodation is a time one, we probably don't need to do something separate. But if there are other kinds of accommodations, let me know. Among the handouts today was, I hope this is the right one. This is fairly vivid. This is also available in color online if you want it. It's just a reminder that there are office hours or discussion times sprinkled across the week for you to take advantage of. We did try in advance to find out what times would be the most useful, and laying it out this way lets you identify the time slots that fit into your schedule. There are ones before class, there are ones after class, there are ones on every day of the week, there are ones in the morning, there are ones in the afternoon. Hopefully there'll be some combination. If it turns out that these start to be filled up too much, we will make sure that there are more hours, but at the moment they're not overutilized. So we would hope that you will find a time that works to get help if you need it. So far, the first two chapters have had quite a bit of review material in them, though I can't heighten too much the idea that you really need to have those functional groups and the comfort with pH and pKa solid. But we're going to be getting into material that is more new and hopefully extends your horizons further. So be current on what we've done so far. If you have been taking kind of an easy approach to it so far, you may want to step up the pace a little bit. In addition to the times that are formally listed there, you can also arrange a time through my secretary for an appointment. I have her arrange all the appointments because if we both do it, we tend to collide. So use the information on contacting her if you want to make an appointment separately. Are there any other, maybe I'll just point this out too. We're actually coming closer to an exam than it seems possible. So it's barely 2 weeks away, just a little over 2 weeks away. So we're going to be working towards that and making sure that you're ready for that. In light of all of that, are there any organizational questions or course organization questions that people have? Are there any questions about Chapter 1 or Chapter 2 you'd like to raise before we go on to Chapter 3? Okay. So let's go on to DNA. DNA is good stuff. We've looked at that. Let me get my pointer hot. I wish they made that a little easier. Okay. So the structure of this first part of the textbook is to explore the chemical properties of the subunits and then the larger biological structures that are made out of that. And it's broken down by the major categories. We're going to start with the nucleic acids, especially for DNA. But the general strategy of looking at the properties of the building blocks and the properties of the larger structures will hold together. DNA is a great place to begin, partly because it is, in fact, the genetic material of all life on Earth today. And we're going to be looking in this chapter at the building blocks, the monomers that go into that. We're going to be looking at how the polymer of DNA behaves when you combine that into a polymer and have now just the residues of the previous things. We'll be comparing a little bit the DNA to the RNA a lot of similarities, some differences. We'll be looking at how that structure permits folding and unfolding, but in the case of DNA, we call that melting and annealing. We'll be looking at information flow, and there's some surprising properties there, and a little bit of what now is possible with DNA that was not possible 10 or 20 years ago. This pair of micrographs here should give you a sense of just how important DNA can be. So this is a comparison here on the top of a histological section of lung in a normal individual, very pretty with all the nuclei lined up. This is a section from the exact same tissue from a patient who has cystic fibrosis. And some of you may know someone in your lives who has cystic fibrosis. This difference between here and there is the difference of a single nucleotide. One nucleotide out of this much DNA has consequences throughout the body for people who have cystic fibrosis. So DNA is a remarkably powerful molecule with great capacity both for construction and destruction, and we'll be looking at both of those. I'd like to just touch a little bit on sort of the background of how we got to where we are with DNA. Gregor Mendel was a monk in Vienna who had the insight that inheritance could be described as if little units were passed down from parents to children. And Mendel's insight was the basis for the history of genetics. It's really quite a long time ago now, 1865, just as the Civil War was ending. Almost the same time, a German investigator called Miescher realized that there were nucleic acids, things that were chemically characteristic of the nucleus of cells. Remember we talked about prokaryotes and eukaryotes, and the eukaryotes have the nuclei. The nucleic acids were characteristic of the nucleus. The way they isolated it then, they tended to look like kind of simple molecules, which of course we know they aren't now. It was the turn of the 20th century that the connection was made between the functional unit of a gene that controlled inheritance and the physical unit of a chromosome, something you could see and look at and manipulate. And it was only in the middle of the 20th century that Oswald Avery was able to demonstrate absolutely conclusively that the genetic material, the stuff that genes, the units of inheritance, are made out of, was DNA. He did this in a series of 3 papers that are incredibly elegant and stand as a monument to the right way to do science, a perfectly beautiful set of papers. Shortly after Avery was the classic Hershey-Chase experiment, which confirmed the identity between genes and DNA in phage. We'll talk about that just a little bit. Just a little later, Erwin Chargaff made an observation that he didn't know what it meant, but it looked like it should mean something. And sometimes in science you have to hold on to those. What he found was that when you look at the DNA from many different species, from sequoias and porcupines and giraffes and George Bush, the composition varies. There can be different amounts of A and different amounts of G and so forth. But the matchings up, the amount of A and the amount of T was always equal, and the amount of G and the amount of C was always equal. And that was one of these it sort of looked like numerology, but it looked like it meant something. And then in 1953, sort of a pair of breakthroughs, Rosalind Franklin proposed a model of a double helix for the structure of DNA, and we'll look a little bit at what her experiments showed. And then Crick had the insight of the base pairing that generated the specificity of the helix. So quite an exciting decade there. It's really quite extraordinary that we can actually look at chromosomes. So if you capture cells during mitosis, the genetic material in the cells is compacted, and you can display it in various ways that allow you to actually examine their organization in the light microscope. This particular set of chromosomes is from amniocentesis, where they have stained it in a way that allows them to distinguish regions that are more and less active, and it allows for a more precise count of the chromosomes present. But we're capable of doing things even much more refined than that now. These are examples of what are called painted chromosomes. You can use a sorter to separate the chromosomes of different sizes and generate probes that are specific to chromosome 1, to chromosome 2, and so on, and end up with an image like this, where each chromosome has a dye associated with it that is unique to that chromosome. So you can uniquely mark every one of the 23 that human beings have. And when you do that in a sample like this, I think this is one of the ones that has, so this is a rearrangement there where a little bit of a different chromosome has been moved over into a location where it's not supposed to be. So what was the Hershey-Chase experiment? You remember I said that Avery had made the claim that DNA was the genetic material in bacteria. This was a very simple and a very elegant experiment. The picture you see here is not a moon lander. This is a bacteriophage, and the landscape it lands on is the wall of a bacterium. You can label that virus by growing it in a medium that has radioisotopes of various kinds. And so if you grow it in a medium that has radioactive phosphorus, we're going to see that the principal place where phosphorus is in DNA. Or you can grow it in radioactive sulfur, and the principal place for that is in protein. And this is a virus that's going to inject its genes into the bacterial cell, but it's not going to enter itself. Only the genes will be shot inside. So you take this radioactive virus, you mix it with bacteria. You want to separate the virus from the bacteria, and they used an extremely simple method. They put it in a Waring blender, and they allowed them to sit together for a certain time, and then they hit the blend button. And the shear tore off the viruses. And now you could ask, where's the sulfur and where's the phosphorus? And the phosphorus was still in the outside soluble part, and the phosphorus with the DNA was inside the bacteria. Hershey and Chase loved this so much that they repeated the experiment something like 15 times. And the phrase Hershey heaven is when you're repeating an experiment that you know will work. So what's going on at a more detailed level with the composition of DNA? In the genetic material of everything that's alive today, there are only 5 bases present. There are 2 that are called purines. So you have adenine and guanine that are purines. We're going to see how it's similar to a purine ring. And there are 3 pyrimidines, thymine, uracil, and cytosine. And these are bases because they are capable of being protonated and so on, but we're representing them here in the unprotonated form. So those are the bases that are part of nucleic acids. If you take a base and the correct sugar, you now have a nucleoside, os for the sugar. Whenever you see os, you should be suspicious there's a sugar involved. And this is showing you some examples of that. Here's cytidine. So if this is cytosine, in this case, it's the base, not a sugar. But cytidine is the cytosine on a ribose. Here's a ribose. And here's a uridine on the ribose. That oxygen is the oxygen that will be gone in deoxyribonucleic acid. So either of these would be nucleosides. And then if you phosphorylate the nucleoside, you have a nucleotide. I can commend the CD that comes in your textbooks. It has very nice demonstrations of the figures and so forth involved here. And here's looking at that in just a little more detail. Here is a purine ring showing you the numbering if you ever need to refer to that. I'm not going to ask you to use those numbering, but just to show you where they are. And you can see that both adenine and guanine share that central ring organization, but that they differ in what's hanging off of it. So in the case of adenine, there's an additional amine group hanging here. In the case of guanine, there's an additional amine group hanging down there and a carbonyl oxygen stuck up there. But you can see the fundamental relationship to the purine ring. Pyrimidine, this is pyrimidine here. Some of you may have used pyrimidine as a solvent in organic chemistry, is the relational name for the other 3 bases. Here's cytosine, modified here with an extra ammonia there and a carbonyl oxygen hanging off. Here's thymine, which you can see has the oxygen, and in this case, oxygen there, and a methyl. The uracil has the 2 oxygens of thymine, but not the methyl. So thymine and uracil have the same capacity for hydrogen bonding, but they differ in the presence of that methyl group. Whereas cytosine has quite different potential for hydrogen, or substantially different potential for hydrogen bonding. It seems odd that the purines are the bigger rings and the pyrimidines are the smaller ones, but that's the way it is. So just remember, the short name is for the big rings, the long name is for the small rings. So let's take a little more look at the nucleosides, and we have to take a little bit of a tiptoe into sugar chemistry to do this. Here is a representation of a ribose ring, and ribose, like a number of the larger sugars, can form a ring. Some of the sugars form rings or linear compounds, some only form one or the other. Certainly ribose is the one that is frequently found as a ring. And the numbering then starts over again for the ribose. So it's 1', 2', 3', 4', 5'. Those are the carbons that are being numbered. And the base is attached at the 1' position. The phosphate is going to be attached either at the 5' or a phosphodiester back to the 3'. 2' deoxyribose has the 2' oxygen gone. So deoxyribose, if that's all you say, is probably a 2' deoxyribose, and it'd be the sugar part in a DNA molecule. And again, if you've got the base and a sugar, that's a nucleoside. Questions so far? To make the nucleotides, we now just make a phosphodiester bond. You may remember that from the list of groups. So here's the 5' sugar on the ribose ring with 1 phosphate. That's adenosine monophosphate. Here's guanine up there with 2, so it's a guanosine diphosphate. Here is cytidine triphosphate. So now what we have is hanging off of one end of the molecule, a nitrogen-rich thing that's not all that polar, and hanging off the other end of the molecule is this, phosphate, often polymer, that's very charged and so forth. So it's kind of a yin-yang molecule just in its own right. Now, the nucleotides are the building blocks, the monomers, that are going to be used to make the polymer of a nucleic acid. You need more than one nucleotide to make a nucleic acid. And the way in which those are found in normally occurring nucleic acids is in the form of a phosphodiester bond. So this here is the phosphate group, but it's in an ester bond to this sugar. So that's an ester bond. And it's in an ester bond to this sugar. And we say it's a 3', 5' phosphodiester bond because it's running between the 3' carbon of the ribose on one member of the polymer and the 5' carbon of the next one in the chain. And so it goes up and down. One chunk within it like this is a residue because it's not a nucleotide anymore. It's really just a residue. People will occasionally call it a nucleotide, or you can call it the base. In fact, there's a water molecule that's been taken out along the way. We'll see that quite a bit later in the semester. Is this a molecule of DNA or RNA? DNA, right, because it's missing the 2' oxygen. You'll also notice that it's a polar compound. That is, the ends of it are different. This end of it has a 5' end with a phosphate hanging off of it. Even if you took the phosphate off of it, it would still have a 5' carbon up there. This end of the molecule, at the moment, has a free 3' end. Under some circumstances, that 3' end might have something else off of it. But there's a directionality inherent to the molecule itself. The molecule is not symmetric end over end. There's a sort of a top and a bottom, if you like to think about it, or a left and a right, depending on how you like to think about it. Now, in the case of DNA, most of the DNA that's found around is present in the form of double strands, in a double helix. And we'll go into the details of the helix in just a little bit. And when you look in a double helix, the bases are hydrogen bonded up and down the helix. And the adenine can form 2 hydrogen bonds, like this. And in this case, between the oxygen and a hydrogen. In this case, between a nitrogen and a hydrogen. So adenine to thymine. Or you can have guanine to cytosine, in which case you have an oxygen, nitrogen, oxygen participating in those hydrogen bonds. It always involves a purine and a pyrimidine. You think about it, it makes sense. Single ring, 2 rings, adds up to 3 rings, 4 rings, 2 rings. Those wouldn't work nearly as well. It's always one purine, one pyrimidine, but it's not just a random one. Adenine will bond to thymine, or will look in RNA to uracil, guanine to cytosine. I would point out to you that we have not exhausted the potential for hydrogen bonding, that these base pairs still have sites that might participate in hydrogen bonding should the occasion occur. And that will turn out to be important when we look at the larger molecule. So this is one of the classical representations of what the structure of the double helix looks like. And we first had significant insight into this from Rosalind Franklin. Rosalind Franklin was an X-ray crystallographer working in England. And based on her X-ray diffraction patterns, she was able to deduce a huge amount of material about the double helix. Based on the pattern alone, she was able to tell that it was an anti-parallel helix. And what does that mean? Well, it means that if you were to look at this strand, here we have the 3' end of that strand sticking up, going down to its 5' end down there, that the partner to it, or the complement to it, is going the opposite direction. Oh, I have a hurt shoulder right now, so opposite direction like that. So that one goes 5' down to a 3' end. They're anti-parallel. She could also tell that the bases were on the interior of the helix and that the phosphate was on the outside. She could tell that it was a right-handed helix. Now, maybe you don't think of things as having right or left hands, but if you were to hold up screws, you can look at them and see that they have a particular way of going. The 2 pedestrian bridges on Riverside come in opposite polarity. One of them is right-handed and one of them is left-handed. And so if you want to sort of practice spotting right-handed helices, take a look at them to get that into your head. She was able to deduce that the pitch of it, that is one full repeat, is 34 angstroms. And she was able to measure that the diameter of the helix was 20 angstroms. Now, this is all for one particular form of DNA. These general features, except for the pitch and the diameter, are going to be more or less true, but we're going to see some variance in a couple of minutes. So this is really quite a lot to know about the structure of DNA. Crick and Watson used her results, in fact, without her permission, to develop a more detailed model in which Crick was able to realize that Chargaff's observation about the rules, that A has to equal T and G equals C, was actually a clue to the structure of the molecule. And so Watson and Crick contributed the base pairing and the way in which it packs in here, that there are 10 base pairs per turn. And out of the structure that they generated that way, an additional feature appeared, which is that the overall structure of the helix is not a smooth cylinder, but it has grooves in it. There's a wide groove called the major groove and a narrow groove called the minor groove. What you see in a DNA molecule will vary with how you represent it. You remember I mentioned that the CD allows you to look at things with different representations. In this particular picture, the diagram is trying to draw your attention to the phosphate backbone, those phosphodiester bonds that run up and down the length of the double helix. And so you can see here the high density of phosphorus and oxygen out there, and negative charges. There's negative charges of it. This representation is trying to draw your eyes to the base pairing, the fact that you have always the same things base paired, blue to yellow or red to green, the fact that the bases aren't quite flat. A lot of times casual people draw them flat, but in fact they're a bit tipped. And so that helps you to sort of see the relationship of the base pairs. And then in this space filling diagram, which is using the same colors as over here, but now is giving van der Waals surface, it's much clearer that there's this major groove here and the minor groove there. Now you may remember I said that we had not used up all of the hydrogen bonding potential of the bases in the base pair. The major groove and the minor groove expose those potential hydrogen bonding sites to the environment. And so what we're going to find is that the sort of Velcro of the major groove and the minor groove allow proteins to interact in ways that are sequence specific in some cases. And that's part of how they managed to pull that trick off. Questions? Okay. So far, in talking about the double helix, I've been talking about a double helix of DNA. It turns out that if you have an RNA molecule that's long enough, so a 5' to 3' polymer of RNA, it is also capable of folding up in many cases. In many cases, the RNA molecules you find biologically are single-stranded, that they don't have a complement lying around. But because of their sequence, they are capable of forming base pairs within that strand. And so while the double helix of DNA is usually between two separate molecules, many RNA molecules have base pairing within themselves. And this is just one example of one very classical one. This is a transfer RNA molecule. And you can see it has a very complex and intricate shape, kind of like a boomerang. And the exact structure, the complex structure it has, in fact, reflects the function it has. We'll see more about that when we get into protein synthesis. But that the long, skinny tail out there and the kind of probe at that end, all of those features are functionally important. So DNA can also hybridize to RNA. The conditions under which that happens are variable. When DNA is hybridized to RNA, the overall helical structure is going to be wider and more compact, and the bases are going to be more severely tipped. When we look at a DNA-RNA hybrid, the general features of the DNA-RNA hybrid resemble a second form of DNA double helix. The first form we talked about, the most common one that's the most abundant in our cells and living things, is called the B form. The A form of DNA, it can also be formed, and it resembles these DNA-RNA hybrids. It is also possible for RNA to make double helices with itself with two molecules, and we most commonly see this in RNA double-stranded viruses. So there are viruses that carry along its genome as a double helix of RNA. So these pictures are meant to give you a sense of the other two major forms of DNA double helices. We've been talking so far principally about the bDNA here. And this is in a space-filling diagram. This is a slightly different way of representing it, sort of a hybrid ribbon base pairing diagram. But there are two other forms of DNA that are easily analyzed and are turning out to be relevant in one way or another. The aDNA is another right-handed helix. It's wider, it's squatter, and as I said, it resembles RNA-DNA hybrids. But this is in this case, this is just a DNA-DNA. There's a third form that double-stranded DNA can take on that's quite different, and the observations of this third form is quite a bit more recent than the A and the B forms. Rosalind Franklin observed both the A and the B forms back in the 50s. The third form is called zDNA, and zDNA is quite a bit different from the, let me see if that's any better on your handout. Let's see. Actually, it's not too bad on your handout. Somehow or other, it's darker on the screen. It's actually not too bad up there either. Okay. 3 different representations. zDNA still has base pairing. Those base pairs are largely on the inside. The phosphate's largely on the outside. zDNA is a left-handed helix. And it's a narrower helix with a longer distance between repeats. So you can see quite a substantial difference between the zDNA and the A and B forms. You can see on this sort of cross-section of the zDNA that the bases are a little more exposed to the outside than they are in the A and the B form. And there's some evidence growing to suggest that zDNA may be more abundant near control regions, and that controlling flipping DNA from the B form to the Z form and back may be one of the things involved in some forms of switching. It is not just a laboratory phenomenon. It does seem to occur in real live organisms. There we go. Questions? Okay. And these give you some specific numbers on that. I'm not expecting you to remember them. Don't worry about that. But it does give you a sense of how different things can be. The aDNA has a tilt of 20 degrees, which is quite a bit more than the others. This is pointing out that the zDNA actually has the opposite rotation from the other two, and that the zDNA is left-handed. If DNA stayed in that double helix, all the time, and was just that beautiful kind of barber pole for all of its existence, it would be pretty useless. We need to get at the sequence of the DNA for it to be any use for anything. And so that means we have to be able to melt DNA and reanneal it. And we're going to look a little bit at that and how it happens and why it isn't happening all the time and so on. The hydrogen bonding between the base pairs allows for smooth packing. That is, the fact that A always base pairs to T and G always base pairs to C means you always get roughly the same size across of base pairs. So it allows for a helix that has an essentially constant diameter going up and down. But the hydrogen bonds that the base pairs are making to each other would have been hydrogen bonds to the water if they weren't in the double helix. So it is not the hydrogen bonding that drives the formation of the double helix in the sense of being in a helix versus being free-floating. The principal force for the stacking comes from interactions between the aromatic rings, that they have an aromatic character that allows the two to stack against each other like a stack of dishes in your cabinet. So this is something more active, so to speak, than a hydrophobic interaction. That in the case of hydrophobic interactions, the hydrophobic molecules don't really interact with each other much. They just don't interact with water. In the case of stacking interactions, the base pairs are interacting with the one above and below them. It turns out that a GC pair is more stable to heating than an AT pair. But that greater stability is not because there's 3 hydrogen bonds instead of 2. It's because of the character of the particular nucleotide pair and its stacking interactions with other nucleotide pairs. There is some contribution from hydrophobic effect, but that's not the major driving force. So what does this look like? We're about to hit another curve. The first curve we saw was a titration curve. We're frequently going to see this, that the midpoint of a curve has interesting properties. So looking at midpoints is one of the things we'll be doing during the semester. In general, I want you to have a good qualitative grasp of things. I will not ask you any questions that need a calculator, but you should be able to get a qualitative answer to questions like this. So if we start off with a solution that's all double helices, and we start to heat that solution up, so here we're raising the temperature as we go along, we can monitor whether or not that's in a double helix or not by looking at absorbance at 260 nanometers. The absorbance at 260 nanometers is, relatively speaking, lower when the bases are base paired to each other and stacked together. But as we warm it, we're going to see a slow rise in absorbance, then a more rapid rise, and then that'll taper off. So when we start to see a rise in the absorbance, some of those strands are starting to melt apart. And when we get the maximum, we're going to get all the strands now are separate from each other as melted free molecules. There's a midpoint to this curve that's halfway between the top and the bottom, and we call that the melting temperature. It isn't something that happens instantaneously. It doesn't all go at once at the melting temperature. The melting temperature is the temperature which half of them are melted and half of them aren't. And we'll encounter that again a lot, that these points of inflection are halfway points. The temperature it takes to melt something is itself information about the DNA. One of the things that can affect the melting temperature is the GC content. The richer something is in GC, the more higher the melting temperature. When you look at the DNA from the organisms that live in boiling water, they're very GC rich. It's also can be affected by the presence of repetitive sequences. And we're going to see that we have in our DNA a surprising amount of repetitive DNA. Now, once you melt this, so here we've diagramized the melting here. We've got them as separate strands. The question is, what happens if you cool it back down? And what happens will vary with how you do that. Any given collection of DNA molecules may have more than one potential interaction. If you cool this solution down in a moderate way and not too far and not too fast, you can get complete reannealing where you get back the original double helix. So the information of the double helix is entirely intrinsic to the helix itself. It does not require a living organism around to reassemble it, which is itself an point of information. But the fact that there are other potential matchups that could happen mean that if you take that solution and you cool it rapidly, you can get mismatches along the way. Regions that are a bit similar can match up, but not to the right part. This is still a reversible process, and if you were to rewarm this toward but not to the melting temperature and give it enough time, you can find them. There'll be little patches that melt, and then when they bounce around, they find another one, so you can get it to reanneal again. And this property is one that we can take advantage of in the laboratory. This capacity of sequences to match up to the sequence that's like them has proven to be a very powerful tool. So we can use short sequences of DNA as a way to pick out of a wide variety of other things specific sequences. So if we have here a variety of samples and we immobilize them on some medium, the medium here might be nitrocellulose, it might be something that can covalently attach them and so on. Something that binds them there in a stable way, but with still the capacity to interact through their bases. If we take a radioactive probe that has some specific sequence of interest, we can then soak that filter or that whatever in the radioactive probe. It will be present everywhere, but the only place where it will stay is the place where it's complementary. And so we can let it bind, we can wash it away, and then visualize it through X-ray, through phosphorescence, through a variety of means. Now, which of these the probe will bind to can be tweaked up and down. Since the melting temperature is one of the variables that controls hybridization, changing the melting temperature will make your probe more or less specific for the sequence that it's looking at. The salt concentrations will affect the specificity with which it binds, or whether it is promiscuous and binds only mildly homologous sequences and so on. We've also developed methods in which this detection has been used through fluorescence, where you can have probes that actually have a dye on them rather than radioactivity. So let's think for a little bit about the DNA not as a structure, but in terms of its function. All that we've talked about so far is the structure of DNA, its helical character, the bases, and so on. But structure always reflects function in biology. Maybe the only place it does, but that's important. And so what I'd like to do now is to sort of walk you through why the particular structure of DNA can perform the functions that it does perform. Francis Crick articulated what he called the central dogma of molecular biology. And that was that information could flow from DNA to DNA, when we have replication, when cells divide and so forth. That information can flow from DNA to RNA, when we make new RNA molecules. And that information can flow from RNA to protein, but not directly. It takes some kind of an adaptation for it. So he emphasized the one-way nature of most information flow. With time, since Crick articulated this, we have found some minor variations on that. There are RNA viruses that just do away with DNA entirely. They just take advantage of RNA for both replication and for coding for proteins. We know now that there is an enzyme that can make DNA from RNA, we call reverse transcriptase. But these are minor variations on the central theme. The vast majority of life goes through the central dogma. All of this is possible because of complementarity. Because DNA has this structure of being a complementary helix. That is, if I give you one strand of double-stranded DNA, you can predict with absolute certainty what the opposite strand should be if you take into account that anti-parallel character of things. The two strands are complementary to each other. They're not identical, they're complementary. And because of that, you can use those strands to generate new strands that are themselves complementary to the original strand. So the complementarity underlies the central dogma. I like this diagram of complementarity during transcription because I think it helps keep straight the character of the strands during transcription. DNA is double-stranded. RNA transcription usually uses just one of those two strands. We'll see a few cases where it may use both. But even if it uses both, what's said here is still true. That in the unwound state, you can't make RNA off of the DNA because the base pairs are all tied up with each other. So you have to open up the DNA, you have to unwind it to some extent, and the RNA polymerase will move along one strand of the two. And the strand that is being used is the template strand, which is complementary to the growing RNA chain, which is synthesized from its 5' end to its 3' end. And then the opposite strand, the one that's complementary to the template, is often called the coding strand because its nucleotide sequence is similar to the nucleotide sequence of the mRNA, with the exception of having U's for T's. And then after that bit of DNA has been transcribed, the DNA is going to close up and reform a helix. But even knowing that is not enough. In order to use the complementarity to make protein, we have to have some kind of rule for how the sequence of the DNA and in turn the RNA gets turned into a sequence of protein. Because the nucleic acids, both the DNA and the RNA, are made of nucleotides. The protein's made of amino acids. We have 4 bases in either RNA or DNA. We have 20 amino acids. There's got to be something going on in order to turn the sequence information of the nucleic acids into proteins. And this was another insight of Francis Crick's, which is that there is a triplet code by which we can turn nucleic acids into amino acids. So if we look here at a small stretch of DNA, here is a messenger RNA that's been coded. You can see that it is complementary to this strand. So this was the template strand. This is called the coding strand, which has same except for the U's and the T's. And this is then going to be read off by transfer RNAs that can match up to it. In principle, it could have taken many possible different forms. Maybe it could have been 2 bases at a time. Maybe it was 4 bases at a time. The use of 3 bases at a time, here's these 3 are going to code for leucine. These 3 are going to code for serine, and so on, gives us certain properties to the translation process that are very useful. It's not overlapping. You use these 3 and then the next 3. That didn't have to be true, but the fact that it is means we have much more freedom about amino acids. We call 3 bases a codon. So 3 bases is a codon, and 1 codon will encode 1 amino acid. And this representation of the genetic code is one that allows you to see some additional properties of the genetic code in kind of an interesting way. So the center is the first base of the codon, the next one is the second, and then the third. So what you can see here is that if your codon starts with U, you can have U, C, and then whatever's in that third position, it'll be a serine. So UCC, UCG, they're all serines. If you look around this wheel, you can see here's a stop codon here. Tryptophan has one. Leucine has a bunch. Proline has a bunch. Alanine has a bunch. Serine has a couple. Arginine has a couple, and so on. We say that the genetic code is unambiguous, meaning if I give you a DNA sequence or if I give you an RNA sequence, there is one and only one protein sequence that can come out of that. It is unambiguous what I'm coding for. However, the code is redundant. That is, it has more than one way to say serine. It has more than one way to say proline. They all mean one thing and one thing only, but there are several ways to say it. It didn't have to be that way. It could have been that we had a three-base codon, and we only used 20 of them plus some start and stops, and didn't use half the codons because there are 64 possible codons. So the code is redundant. But it has a third feature that was not obvious a third feature that was not obvious to Crick, and it was one that could not be really known until people thought about it a little more, which is that we have a resilient code. And this is kind of an interesting property. And there's a paper describing this that I'll make sure I put on icon for you as well. If we look at the codons that code for a particular amino acid, in the triplet code that we have, the change of any one base is quite likely not to change the sequence of the protein, given the fact that there are 64 bases. And so this is a sort of a diagram trying to convey that of how resilient this code is to changes of accidents or mutations or whatever. And that you have things that are somewhat similar to each other having codons that are also somewhat similar to each other, so that if you have a mutation, you either end up with the same amino acid or at least an amino acid that's conservative. We take this for granted, but it is not an automatic property of the genetic code. Someone did Monte Carlo simulations to look at all the possible genetic codes, and only 1 in a million was as beautiful, as resilient as the genetic code that we actually have. So that is a truly impressive property of the genetic code. And with that moment of awe, I will say have a good weekend, and I'll see you on Monday.